blood collection on other site. Centrifuge in order to separate serum from blood. Preparation of washed red blood cells. Transfer to drops of red cells to a labeled test tube. Add as much volume of NS until the mixture fills 2/3 of the tube. Cover with parafilm. Mix it by gentle inversion 3 to 5 times. This process is known as washing. Centrifuge the samples for 2 minutes at 3,400 revolutions per minute. Pipette and discard the supernatant on a waste bottle. Make sure the packed red cells are not disturbed. Repeat steps 2 to 5 up to 3 times. In cross-matching using gel method first, dilute 50 ml donor's red cell with 1 ml DG gel salt to prepare 1% red cell suspension. Then dispense 10 ml of donor's red cell suspension to the corresponding well. And dispense 25 ml of the patient's serum plasma into the same well. Incubate for 15 minutes at 37 C. Then centrifuge for 9 minutes. For the interpretation, based on the fourth well from left there is presence of a red cell button at the bottom of the micro tube. It is recorded as compatible. In a BO forward typing the P7 and P8 has the presence of agglutination, while P6 don't have. P6 is a mix of 1 drop of anti-A and 1 drop of patiential suspension. P7 is a mix of 1 drop of anti-B and 1 drop of patient cell suspension. P8 is a mix of 1 drop anti-D and 1 drop of patient cell suspension. Therefore, the patient is B positive. In a BO reverse typing the P9 has the presence of agglutination, while P10 don't have. It confirms that the patient is B positive. In a BO forward typing for the donor the D7 and D8 has the presence of agglutination, while D6 don't have. Therefore, the donor is B positive. In a BO reverse typing the P9 has the presence of agglutination, while P10 don't have. It confirms that the patient is B positive. ABO and RH forward typing slide method. And copper sulfate screening. 1 drop of anti-sera A. 1 drop of anti-sera B. 1 drop of anti-sera D. Copper sulfate screening through capillary puncture. Agglutination in anti-sera B and D. Direct antiglobulin test. In DAT, dispense 1 drop of a 2% to 5% suspension of red cells into a clean, labeled test tube. Wash each tube 3 times with normal saline solution. Then completely decant the final wash. And blot dry using a gauze or paper towel to remove residual saline. Then after that, immediately add 1 drop of anti-human globulin to the cell suspension. Centrifuge for 30 seconds at 3,400 revolutions per minute. The DAT result is negative since no agglutination is observed. A negative DAT result does not necessarily mean that the red cells have no attached globulin molecules. Minor and major cross-matching has no agglutination. Rechecking for the result. The result form.